Microbubbles, tiny but mighty. For Dr. Zhang's Neurobiology course, we are going to be talking about a new method of drug delivery involving microbubbles to pass the blood-brain barrier. But to start, we must first understand what the blood-brain barrier is and why it is so important. The blood-brain barrier is a layer of endothelial cells that surround the blood vessels of the brain and prevents many substances from reaching the brain and spinal cord, while also allowing important nutritional substances to pass, for example, glucose. It does this through very selective tight junctions between endothelial cells. Besides endothelial cells, the blood-brain barrier is made up of four other key parts: pericytes, astrocytes, neuroglial cells, and the basement membrane. All of these cells and tissues work together to dilate, constrict, influence permeability, provide immune responses, and provide structural integrity for the barrier. It is important that we are able to non-invasively pass the blood-brain barrier to deliver vital pharmaceuticals to possibly treat brain cancer, Alzheimer's, Parkinson's, and more brain diseases. Microbubbles are used as tools to deliver nucleic acids for research, carry drugs to targeted regions, and are helpful in medical imaging. Microbubbles are typically between 0.5 micrometers and 10 micrometers. They are filled with gas and can have nanodrugs or dyes attached to the outside. A study in 2017 used Evans blue dye combined with ultrasound to determine the penetrance of microbubbles into the striatum of mice. The dye was present in the medical imaging and thus proved the microbubbles were successful. Other previous experiments have demonstrated that for a given concentration, microbubble size affects both the persistence of the substance in the blood and the extent of the blood-brain barrier opening. The bigger the microbubble is, the more drug concentration it can carry. This makes it seem like the obvious choice for maximum pharmaceutical delivery. However, bigger microbubbles are more likely to be targeted by an immune response. It is estimated that between 80 and 90% of microbubbles will be destroyed or not reach the target membrane. Thus, it is important to find a balance between the size of the microbubble to deliver maximum drug concentration and evading an immune response. Microbubbles are especially useful when combined with ultrasound as they can target very specific tissues to deliver drugs. Ultrasound-induced cavitation disrupts the microbubbles and releases the drug in a specific desired region. This process is seen in the picture. It involves the expansion and compression of the microbubble as a response to the altering ultrasound pressure waves. Ultrasound increases the space between the adjacent primary endothelial cells, allowing the drug molecules on the microbubble surface to penetrate the barrier. This is called sonoporation and happens at higher pressures as the bubble dynamics become active enough to create pores in the membrane. Cell pores that last longer than 120 seconds can lead to cell death due to disruptive homeostasis. This occurs with larger microbubbles, so smaller bubbles are preferred in this regard as they create smaller pores in the cell membrane and can thus close faster. The targeted release of the drug with the ultrasound allows for local delivery and increased uptake of the drug in the desired cells while limiting the activation of distant undesirable targets. One of the biggest limitations of using microbubbles, however, is the transient nature of the bubbles. Following an IV injection, microbubbles have a circulation life of about 10 minutes. This is due to rapid clearance by hepatic Kupffer cells. Using polyethylene glycol as a shell constituent for microbubbles increases their circulation time as it is more effective at evading opsonization. A promising study from 2021 used microbubbles to deliver pharmaceuticals to the brain to help treat Alzheimer's. The drug, brain-derived nerve growth factor, otherwise known as BDNF, is used to slow the progression of neurodegeneration in Alzheimer's disease via promoting neural regeneration and angiogenesis. Neural regeneration involves creating new connections for neurons to replace old damaged ones, thus reversing the effect of Alzheimer's. Angiogenesis is the formation of new blood vessels and is thus important in ensuring proper oxygenation to the brain. This drug can also reduce cell loss as well as increase synaptic regulation and axonal plasticity. However, the BDNF drug cannot cross the blood-brain barrier without injuring brain tissue. Incorporating the drug with ultrasound-targeted microbubble destruction is a non-invasive and specifically selective delivery aid that improves the drug concentration in targeted zones without injuring the brain tissue. These pathways created by microbubbles are reversible and do not cause any cellular damage. In this case study, 32 adult male rats suffering from Alzheimer's were studied. The rats were placed into four groups. the control group, and 3 experimental groups that consisted of various versions of peptides, either BDNF or EGFP. These were prepared with a retrovirus before being exposed to the MRI-focused ultrasound waves. The observations regarding the experimental group showed the blood-brain barrier opening, and the results proved an increase in synaptic density as well as a decrease in the amount of enzyme choline acetyltransferase, specifically the number of choline acetyltransferase positive neurons. Choline acetyltransferase is an enzyme that synthesizes acetylcholine and is found in cholinergic neurons. It is important to note that deficits associated with cholinergic neurotransmission are known to influence many of the psychiatric symptoms associated with Alzheimer's disease due to the disruption of cholinergic input to the cortex. Thus, it showed promising results in the mice models. The image to the left shows the decrease in cholinergic neurons in the hippocampus amongst the experimental groups compared to the control group. The brain tissue is stained for choline acetyltransferase positive neurons, which gives off a rusty brown color as a result. Panel A shows the wild-type group. Panel B shows the effect of Alzheimer's brains being treated with a control peptide, EGFP, via a retrovirus. Panel C shows the effect of an Alzheimer's brain being treated with pro-peptide BDNF via a retrovirus. Panel D shows the effect of an Alzheimer's brain being treated with pro-peptide BDNF via a retrovirus and microbubbles. It is important to note that panels B through D received injections of their treatment through the tail vein, followed by their hippocampus being exposed to sonication via low-frequency focused ultrasonic waves in order to open up the blood-brain barrier. Visually comparing the panels amongst one another, panel A has the most choline acetyltransferase positive neurons compared to panel B, which has the least amount of choline acetyltransferase positive neurons, as this is consistent with neural degeneration in Alzheimer's patients. Focusing specifically on the experimental groups, panel C shows a dramatic increase in the survival of cholinergic neurons that are choline acetyltransferase positive through direct treatment with BDNF, while panel D shows a slight increase in the number of neurons through treatment of non-invasive delivery of BDNF via microbubbles. Graph E shows the average number of choline acetyltransferase positive neurons analyzed within each panel group. This is another figure from the article that shows the blood-brain barrier being disrupted by the microbubbles combined with the BDNF retrovirus and then being exposed to the ultrasound waves. The top 2 photos show an MRI monitoring the blood-brain barrier disruption. The barrier opening is confirmed by the leakage of the MR contrast agent through the blood-brain barrier and into the cortex. The 2 bottom photos confirms the location of the blood-brain barrier being opened through the EB staining of affected area being presented. The conclusion of the study confirms that the interaction between ultrasound waves and microbubbles can efficiently deliver BDNF into the brain in order to prevent neuronal death in Alzheimer's in a non-invasive way. Using microbubbles to treat brain cancer has also shown promising results in mice models. This method could be used to introduce small interfering RNA within cells to achieve gene silencing effects, which results in reduced tumor growth. This could be very useful in cancer treatments by preventing the growth and rapid expansion of the tumor and thus eliminating its pathogenicity. Additionally, the microbubble method enhances delivery of several chemotherapy agents across the blood-brain barrier, including Herceptin. This results in a more targeted cancer therapy as the microbubbles will only penetrate where they are directed to go. This means healthy tissues will not be as affected, and thus this can improve the overall quality of treatment and result in a better outcome for patients. While microbubble experiments involving the blood-brain barrier have not yet made it to the human testing stage, this method shows promising results and can have a major impact on medicine in the upcoming years.